Most current foundation models for brain imaging are built on macroscopic anatomy, relying on T1 or T2 weighted structural scans, or they focus on functional dynamics. But the real biological story happens at the microstructural level. Conventional diffusion, MRI metrics like fractional anisotropy and mean diffusivity are useful, but they fundamentally lack biological specificity. They conflate neurite loss, demyelination, edema, and neuroinflammation into a single scalar value. To actually isolate these distinct tissue compartments, researchers use biophysical models like NODDI, which decomposes the diffusion signal into three parametric maps: the neurite density index, or NDI, which captures neurite packing, the orientation dispersion index, or ODI, which measures fiber coherence, and the free water fraction, or FWF, which tracks extracellular fluid. These three maps provide an incredibly rich substrate for predictive modeling. They reveal systematic microstructural trajectories across aging, provide mechanistically grounded sensitivity to cerebrovascular injury, and detect early neurodegenerative alterations that macroscopic scans completely miss. But here's the computational bottleneck. How do you effectively fuse these three complex, three-dimensional spatial maps without re-entangling the very biophysical processes NODDI was explicitly designed to decouple? Standard multimodal fusion strategies simply collapse different modalities into a monolithic latent space. You lose interpretability, and you risk the model learning spurious correlations instead of genuine tissue interactions. To solve this, researchers introduced BrainFIBRE, the first foundation model dedicated to brain tissue microstructure. Instead of treating the three NODDI maps as a single concatenated input, BrainFIBRE treats them as distinct modalities and explicitly models how they interact. The core innovation here is a framework called Self-supervised partial information decomposition, or SPID. To understand why SPID is necessary, we need to look at partial information decomposition, or PID. In information theory, PID breaks down the total information that multiple sources carry about a target into three components: uniqueness, redundancy, and synergy. Uniqueness isolates what each modality contributes on its own. Redundancy captures information shared across modalities. And synergy reveals information that only emerges when you observe the modalities jointly. Previous attempts to operationalize PID for machine learning relied on supervised label agreement between classifiers. That approach strictly ties the decomposition to downstream task quality and completely precludes the large-scale, label-free pretraining that foundation models require. BrainFIBRE bridges this gap by extending PID into a purely self-supervised regime. The architecture is built around a mixture of experts' design. Three unimodal 3D vision transformers encode each NODDI map into separate embeddings. These embeddings are then routed to 5 specialized experts: 3 uniqueness experts, 1 redundancy expert, and 1 synergy expert. Each expert is tasked with isolating its designated information component. But how do you train these experts without any ground truth labels? The answer lies in a paradigm they call Counterfactual Candidate Construction, or CCC. Standard spatial augmentations preserve the natural alignment between the three maps, which means the model is never forced to distinguish between modality-specific signals and the natural co-occurrence of the compartments. CCC deliberately disrupts this alignment to generate contrastive teaching signals. For every participant in a batch, the framework generates 2 augmented views. One view serves as the anchor, while the other is systematically perturbed through 2 operations: modality dropout and modality swapping. Modality dropout replaces a specific NODDI map with Gaussian noise, simulating its absence. Modality swap replaces a modality with the exact same map from a different participant in the batch. Because human brains share highly conserved macroscopic anatomy, swapping a modality yields a counterfactual that looks anatomically valid, but is fundamentally microstructurally mismatched. By forcing the model to distinguish these subtle disruptions, CCC provides the exact contrastive signal needed to disentangle information in a label-free manner. These perturbations feed into expert conditional interaction rules. For example, the uniqueness expert for ODI should be invariant to changes in NDI and FWF, but highly sensitive to perturbations in ODI itself. So dropping or swapping NDI and FWF are treated as positive contrastive examples, while dropping or swapping ODI are treated as negatives. The redundancy expert, on the other hand, should remain robust to missing partial information, making all dropout candidates positive, but it should break down when cross-participant swaps occur, making all swaps negative. The synergy expert is the most restrictive. It only receives the unperturbed base candidate as positive, because any dropout or swap destroys the emergent multimodal interaction it's designed to capture. With these rules in place, the model is optimized using a multi-positive symmetric InfoNCE loss. This loss maximizes the similarity between the anchor embedding and its positive counterfactual candidates while pushing away negatives and all other participants in the batch. To learn robust, participant-specific representations, a global contrastive loss is applied to the final fused embeddings. Because training mixture of experts models in a self-supervised setting is notoriously prone to representation collapse, where the model trivially routes everything to a single expert, 2 auxiliary regularization terms are introduced. A balance loss minimizes the KL divergence between the batch-averaged expert assignment probabilities and a uniform distribution, preventing global expert collapse. An entropy loss encourages soft probabilistic assignments, ensuring each participant integrates information from multiple experts rather than relying on deterministic, one-hot routing. The entire framework was pre-trained on NODDI-derived microstructure maps from over 55,000 participants in the UK Biobank. When evaluated on a comprehensive suite of downstream tasks, BrainFIBRE achieves state-of-the-art performance across diverse prediction benchmarks. It outperforms unimodal baselines, fully supervised multimodal models, and existing self-supervised foundation models on demographic prediction, future hippocampal atrophy, processing speed, and white matter hyperintensity volume. Crucially, this performance isn't limited to the training cohort. The model generalizes strongly to the independent human connectome project in aging dataset, and it maintains robust predictive utility on the Singer cohort, an ethnically distinct Asian population enriched for cardiometabolic risk. This cross-cohort resilience suggests the model learns fundamental, transferable microstructural features rather than dataset-specific biases. But the true power of this approach becomes apparent when you look at interpretability. The learned representations yield neurobiologically meaningful patterns that align with established neuropathology. By analyzing the distribution of expert weights across different tasks, you can see exactly how the brain's microstructural compartments interact under different conditions. For age prediction, the synergy expert consistently carries the highest weight, indicating that aging is best captured by emergent information fused jointly from all three NODDI maps. However, the uniqueness contributions shift meaningfully. FWF dominates age prediction, reflecting widespread extracellular water expansion in aging, while ODI dominates hippocampal atrophy prediction, pointing to alterations in neurite dispersion as a key marker of regional neurodegeneration. In the Asian cohort, the interaction patterns adapt to the underlying disease risk profile. Age prediction remains synergy-dominated, but shows a stronger ODI uniqueness contribution, suggesting that changes in fiber architecture and dendritic organization provide additional aging-relevant signal in populations with higher cardiometabolic burden. For white matter hyperintensity volume prediction, the pattern shifts dramatically. FWF uniqueness becomes the primary contributor, followed by redundancy and synergy with minimal reliance on ODI. This aligns perfectly with established findings that WMH burden is driven predominantly by extracellular water expansion, rather than intracellular structural changes. Spatial attention patterns further validate the model's biological grounding. The uniqueness experts reveal modality-specific cortical signatures. NDI localizes to the medial temporal cortex, ODI maps to posterior regions, and FWF spans the broadest cortical extent. In white matter, age prediction is driven by limbic and association tracts, with synergy and redundancy experts consistently highlighting the cingulum as a hub of multifactorial microstructural aging. These spatial signatures match decades of lifespan diffusion studies, confirming that BrainFIBRE isn't just learning statistical correlations, it's learning voxel-level biophysical correspondences. By treating neurite density, orientation dispersion, and free water fraction as distinct modalities, and explicitly modeling their unique, redundant, and synergistic interactions, BrainFIBRE establishes a versatile, biophysically grounded foundation for neuroimaging analysis. It moves beyond macroscopic black box fusion and provides a principled, self-supervised framework for understanding how different tissue compartments interact across health, aging, and disease. The architecture, the self-supervised decomposition strategy, and the cross-cohort validation all point to a new direction for computational neuroimaging, one where representation learning is explicitly tied to the underlying biophysics of the tissue it's analyzing. The code and pre-trained weights are publicly available, providing a ready-to-use foundation for researchers looking to probe brain microstructure at scale. If you're working on diffusion MRI, neurodegenerative modeling, or cross-population generalization, this framework offers a rigorous, interpretable starting point that bridges information theory, self-supervised learning, and computational neuroscience.